replicated DNA getting ready to go through meiosis has the genotype big D, big D, big B, big H, big A, little a, big D, big H, big A, big B, big B. So when this cell goes through meiosis, can you see this? The daughter cells will be either big B or little b, big H or little h, big A or little a, and they will always have the big D allele because that's the only allele mom has, so they can't have any other allele. However, as you can see from the chromosomes, right now, big H and big A will always end up in the same daughter cell, and little h and little a will always end up in the same daughter cell. Recombination mixes all of that up. The chromatids get a little bit intimate with each other. So as this homologous pair is moving around, getting ready to line up in the middle of the cell, what happens is that pieces are exchanged. So zoom in on this. So the little b actually swaps with the big B. A piece of DNA is exchanged, and now there's a big B where there used to be a little b, and a little b where there used to be a big B, and the same thing happens over here. These chromatids get intimate with each other, and a piece is exchanged, and now there's a big A instead of a little a here, and a little a instead of a big A over here. So now a novel chromosome that never existed before is created. The hairy ear gene, the dominant hairy ear gene, is on the same chromatid as the recessive achondroplasia allele. So now when the cell goes through meiosis and divides twice to form 4 haploid daughter cells that each have one chromosome number 3 and one chromosome number 4, we have a cell with a unique combination of alleles that did not exist in either parent. So every gene is independent of every other gene, with an exception we're not going to talk about today, where if there's a disease gene like achondroplasia, it ends up in a sperm or egg completely randomly and has nothing to do with any other allele of another gene. And that's because of crossing over. So hopefully that helped. Good luck with your labs.